Okay, so we are working on chapter 10, and we got through the first page into the second page, and getting into how a muscle is organized and how it's put together. I had indicated to you guys that we kind of think of a muscle as one large structure, when in fact you have a bunch of individual bundles that are continuously bound together to eventually arrive at the muscle as a whole. And so we're going to talk about those individual parts that make it up. Now, with the individual parts that you have within a muscle, you have the muscle tissue itself, and then you actually have the membranes that are going to be wrapped around the different bundles that you have and holding them together. So as we move through this next section, we're going to be talking about both the muscle tissues that we have as well as the membranes that are going to help hold those bundles together. We're technically on page 2, but I want you guys to take a quick look at page 3 on your packet. On page 3 at the top, you guys have the visual part that goes through this, and I think it would be beneficial for you guys to take a look at the pictures that you have first, and then kind of work through the lecture as we go through it. Another good key would be, if you have your textbook with you, to possibly look at it in the textbook. Again, the color I think does so much more to kind of show all the individual structures, and obviously in your textbook it's going to be much larger than the slides that you have in your packet. So those are my 2 suggestions. If you have your textbook, I would strongly suggest opening up your textbook to chapter 10 and taking a look at those pictures. If you don't have your textbook, at least pause it for a moment, flip over to page 3 and take a look at those images that you have on page 3 before we continue on with the parts of the muscle. Now, with the organization of the muscle tissue, you have 3 different membrane layers that are going to play a large role in taking the individual parts and bundling them together. You have the epimysium, the perimysium, and the endomysium. And the prefixes that you have on these 3 connective tissues is actually an indicator of where they're located. Epi falls in line with the same prefix that you see on the epidermis, telling you that it is the outermost layer. Endo tells you that it is in, so this is always the innermost layer. And then you have the peri, which is going to be in between the 2, and essentially is found in the middle. So this is one of those situations where these prefixes are going to hold true, and no matter where you would see these prefixes, epi is going to always be the outermost, endo is going to be the innermost, and peri, if you have one, is going to be the middle layer. And I say if you have one because sometimes you'll just have the epi and the endo, but like I said, these will show up in other places in the body, and just use that as kind of a way to decode the information that it's trying to relay to you. Now, within the epimysium is going to be a layer of connective tissue that is going to take all of the individual bundles and wrap them together. So the best example that I can give you, if you've ever gone to a restaurant and ordered food to go, whether it be McDonald's, Subway, somewhere like that, they prepare the sandwich, all the individual parts, they put that together, and what do? They take a piece of paper and they wrap it up in that piece of paper to hold it all together. Essentially, that wrapper that you have on the outside is the equivalent of what the epimysium does for the bundles or the individual components that you have with the muscle. So epimysium is going to be the outermost layer, taking all the bundles that you have and holding them together. Then you have the perimysium, and the perimysium is going to be a connective tissue layer that is going to hold individual bundles together. And those bundles are going to be referred to as fascicles. And fascicles are made up of yet even smaller parts known as myofibrils, or your muscle fibers. On the inside of the fascicles, you have the muscle fibers, aka the muscle cells, and these are going to be held together by that connective tissue layer known as endomysium. So kind of working your way smallest to largest, you have these muscle fibers The muscle fibers are all going to be held together within a bundle. That is going to be the endomysium. When you put a bunch of those muscle fibers together and you package them as one bundle, that is referred to as a fascicle, and the perimysium is going to wrap around the fascicle. You take a bunch of the fascicles and lump them together, and now you're going to take the epimysium, wrap that around, and that will complete your entire muscle. Now, some of the other connective tissues that we have involved with the muscle fiber are going to be the tendon and what is known as the aponeuroses. A tendon, we've already indicated, is a structure that is going to hold muscle to bone. And the actual tendon itself is going to arise out of the muscle tissue. So it's not something that is attached or on top of the muscle, but it actually is embedded and part of the muscle fibers themselves. And this is part of where the strength comes from, is that, like I said, it's not attached or on top of. It's actually embedded and part of the muscle tissue itself. It's an extension of the muscle tissue. And it's going to stem from the muscle tissue to a point on the bone and adhere to usually the periosteum. And again, that's going to be another source of strength, having it be directly part of the periosteum, and again, not just attached or adhered to the surface of the bone. An aponeuroses is essentially the equivalent of a tendon, but it's a very large, broad sheet of tendon. We see these in several different locations throughout the body. You have one on the top of your head that connects the frontalis muscle to the occipitalis in the back. You also have another one through your abdominal region to help support and keep your body upright when you're in the standing position. So just a couple of examples of where you would have an aponeuroses, but it's the equivalent of a tendon, it's just on a much larger scale, and so you get a different term for it to indicate that it's a much larger tendon structure. Moving down to the bottom of page 3, it talks about blood vessels and nerves. And muscles are constantly working. 24 hours a day, 7 days a week, they have to be doing something. They don't get to rest. Just because you are sitting in a chair or you're sleeping in your bed and you are not being physically active, your muscles still have to work. And because they are constantly working, it's basically if you were to leave your car running, even if you weren't driving it around and going anywhere. Car's running, the engine's going to have to do work, and you're going to have a fuel source in order to keep that car going. For your muscles, the fuel source that it's going to need in order to be able to continue working is going to be oxygen. So when we talk about blood vessels serving the muscles, you actually have a high number of blood vessels that are going to be taking care of your muscles because they constantly have to be nourished with an oxygen supply. You're also going to have a very good nerve system that is dealing with your muscles, simply because the impulse or the stimuli to cause a muscle contraction is going to come directly from those nerves. And so you are going to have multiple nerves serving a muscle at a variety of different locations, so that when and wherever an impulse is needed for that muscle to contract, the nerves are right there to send that over to the muscle and cause the contraction to happen. Some of the muscle cells that you have, excuse me. The first type of muscle cell that it talks about is a myoblast. And with the myoblast, whenever you see that prefix myo, M-Y-O, that is going to indicate to you that you are dealing with a muscle structure. And the term blast is going to then tell you that it is a new or an immature version of a muscle cell. The muscle cells that you have typically tend to be elongated cells, so they're long and narrow. The reason for having this long, narrow shape is because when a muscle contraction comes in, it's going to come into one specific location within that muscle, and then it has to extend and send that impulse down the length of the muscle to get the entire muscle to contract. and you kind of get a domino effect. So by having these long, narrow muscles, it just helps with that contraction and sending the information from one part of the muscle down to all the other parts of the muscle as well. You also have multiple nuclei in your skeletal muscles, so you find them all over the place. And typically within your skeletal muscles, we see something a little bit different than what we see in other cells. Typically, your nuclei is always going to be held at the center of a cell to offer the nucleus protection. That's where your genetic information is held, so we want to make sure that it's very well protected. In the case of the muscle cells, the nuclei, you have multiple within a single muscle cell, but also it's pushed up against the edge. So we see something very different than what we have in the past, where, like I said, it's held at the core. But now, and this actually is one of the defining characteristics for knowing that you're looking at skeletal muscle, is that the nuclei are going to be up around the edge. They're pushed out to the side and typically are right up against the cell membrane of the muscle cell. Turn over to page 4. On page 4, it starts to get into some of the internal structures that you have within a muscle cell. And a lot of these will overlap and have the same names as your typical body cell or a somatic cell. Some of them are tweaked a little bit to refer or indicate that they are talking very specifically about a muscle cell. So starting on page 4 with the sarcolemma and the transverse tubules. Sarcolemma, very easy way of saying cell membrane. So it's talking about a layer of tissue that is going to envelop the muscle and hold all the contents into one place. You also have the sarcoplasm, aka the cytoplasm. And remember, the sarcoplasm is going to be a gel-like material that is going to fill all extra space. So anything within the muscle that is not filled up by organelles and other structures within the cell is going to be taken up by that sarcoplasm. Easy way to think about it, if you think about the room that you're sitting in right now and you look at all the items that are in the room, chairs, tables, things like that, wherever you're at, any area within that room that is not taken up by tables, chairs, pictures, computers is going to be filled by air, and that's the equivalent of what sarcoplasm does within a cell. You also have the transverse tubules. And the transverse tubules are the structures that will actually dive down into the muscle tissue. Now remember, you have all these bundles that are together, but when a muscle needs to contract, the impulse or the stimuli for that contraction to happen actually needs to be able to get all the way down into the muscle itself so that the entire muscle is going to carry out the contraction. In order to do that, you have to have a network of tubes that are going to dive down into the muscle tissue and be able to relay the chemical that's going to cause the muscle contraction to happen. And that's the job of those transverse tubules. They are actually going to dive down into all of those bundles and weave in and out of the bundles so that it can deliver a neurotransmitter known as acetylcholine, typically abbreviated as ACH, but acetylcholine is the chemical that is going to trigger a muscle contraction to happen. Then you have the myofibrils. Myofibrils are going to run horizontally down a muscle fiber, and the myofibrils are going to be made up of different types of proteins. They're referred to as myofilaments. Again, that prefix myo always indicating that you're dealing something with muscle. And filaments, typically when we talk about filaments, we're talking about protein rods. The two different types of myofilaments that you have within a muscle are going to be known as myosin and actin. And myosin and actin are two different types of protein. Actin is known as the thinner protein, and it actually will appear lighter. So when you actually look at an image of a skeletal muscle, you're going to see this banding pattern known as striations. And the best example I can give you that you have actually seen of this is what you see on a zebra. You have the white stripes, you have the black stripes, they alternate with each other. That is the same setup that you have with a skeletal muscle. You're going to have these alternating light and dark bands. The lighter bands are made up of a protein known as actin. Like I said, they get that lighter color because they're thinner in nature. Myosin is a second type of protein but this protein is much thicker, and so it is going to appear as the darker band within the skeletal muscle tissue. Flip over to page 5. Page 5 talks about the sarcoplasmic reticulum. Sarcoplasmic reticulum is going to be the same equivalent as the endoplasmic reticulum. It's a network of channels that you have running through the muscle cells, and they are going to again deliver materials. When I introduced it with the body cell, I talked about it being like the hallways of a building. And so typically what you have is the sarcoplasmic reticulum is going to run horizontally across the muscle, and the transverse tubules are going to run transversely through the muscle tissue. Now, with the sarcoplasmic reticulum is going to complete the structure known as a triad. And a triad is going to be 3 tubes, hence the name tri. And the center tube is going to be one of those transverse tubules before it dives down into the muscle itself. And then on either side of that, you are going to have these extensions of the sarcoplasmic reticulum known as the cisternae. And the cisternae is essentially going to be the holding tanks for the calcium that you have within your muscles. And so to kind of stair-step it through, you have this nerve that is going to lead right up to the muscle tissue. A little bit of a gap between the nerve and the muscle so that the muscle is not always getting an impulse or the stimuli to contract. But when that nerve receives an impulse that the muscle needs to carry out a contraction, first thing it's going to do, it's going to release a neurotransmitter, which I already introduced, acetylcholine. That acetylcholine is going to diffuse across that small gap between the nerve and the muscle, and once the acetylcholine hits the surface of the muscle tissue, it's going to be taken in to the muscle through the transverse tubules that you have. And once the acetylcholine gets into the muscle tissue, it's going to trigger those cisternae to release the calcium that has been stored in the holding tanks, essentially. So we have the acetylcholine coming over from the nerve, hits the surface of the muscle. That presence of the acetylcholine is going to cause the sarcoplasmic reticulum, those cisternae, to release the calcium that they've been holding on to. And now that is going to trigger the muscle contraction to begin. On the bottom of page 5, more visuals showing you where the cisternae are located, the sarcoplasm, sarcolemma, the myofibrils. So you have just all those different structures that we introduced, and it's showing it to you visually. Again, I would strongly suggest, if you have your textbook, getting your textbook out and taking a look at those structures in color. Turning over to page 6. Page 6 gets into a sarcomere, and a sarcomere is a contractile unit that you have within a muscle. If as a little kid you ever had a pen that was a spring-loaded pen, and most people at some point in their lives have taken away or taken apart a pen that has a spring in it, and you play around with the spring. If you've ever done that, or if you've ever played with a spring in general, essentially that spring is what a sarcomere is and how a sarcomere works. So if you just leave that sarcomere on the table by itself, it's not going to do anything. But if you pick it up and you pinch it between your fingers, you're going to take that sarcomere, you're going to reduce its size, and it's going to become shorter in length. If you let go of the spring and let it sit back on the table again, it will slide back to its original position and its original size, but it's not going to go any further. It will only go back to its original size, and then it will stop. Pick it up and you pinch it again, you're going to shorten it down in size, and you can repeat the process over and over again. That's essentially what a sarcomere does. But within your muscles, you are going to have hundreds to thousands of these sarcomeres lined up end to end. Muscle contraction happens, it's going to cause those sarcomeres to shorten down, just like if you pinched a spring. And it will happen with one, domino effect happens to the neighboring sarcomere, on down the line until you get all 100 of these sarcomeres shortening at once. And on the outside, you see the shortening of all those sarcomeres as a muscle contraction. Now, within the sarcomere, you have several different structures that are going to make it up. And at the bottom of page 6, you have the visual representation of the sarcomere. And it's showing you one individual sarcomere. And keep in mind that for an entire muscle, you would have hundreds to thousands of these lined up end to end. Now, I am going to introduce these, and then we will be finished with our lecture for today. So starting with the A band. The A band is going to be the location of the sarcomere where you have both actin and myosin present. Actin and myosin are those two proteins that I mentioned a little bit earlier, the thick and the thin filaments, light and dark in color. And these are the two proteins that are actually going to be responsible for a muscle contraction and that sarcomere becoming shorter in length. So within a sarcomere, the region where both actin and myosin are found is going to be known as the A band. Now, you also have what is known as the I band, and the I band is going to be where the actin attaches to the end of the sarcomeres. So you have this little contractile unit, and a lot of times when I introduce the sarcomere, I kind of tell people to think of it as a football field. At the end of the football field, you have the end zones, and that tells you where the end of the football field is. When we talk about a sarcomere, the ends of the football field, those end zones, would be what we call Z lines. Now, off of the end zone, what do you have? You have the out of bounds lines, and they are going to be perpendicular or run perpendicular to the end zone. Where the out of bounds lines and the end zone cross each other, that point of attachment is going to be in a sarcomere known as the I band. And that essentially is where the actin protein is going to come in contact with the Z line or the end of the sarcomere. Okay. Now, the myosin is going to be in the middle of a sarcomere, and it's held in place by a protein known as the M line. Okay. Now, the M line holds the myosin in place. The myosin does not move. So when a sarcomere contracts and becomes shorter in length, it's actually due to the actin. And the actin is often referred to as the sliding filament because it's the one that is actually going to shift and move in the sarcomere and cause the sarcomere to become shorter in length. All right, so I know that I introduced a lot of new terms. I am actually going to go back over sarcomeres when we meet in class on Monday. So if that was completely foreign to you, don't worry. I would fully expect it to be. Most people have no background knowledge of muscle parts when they come in. So I wanted to introduce it today, not because I expected you to be done with the lecture and be experts on sarcomeres, but I at least wanted you to hear the terms so that when we come back to it on Monday, you've heard of them, you have a little bit of an explanation, a little bit hopefully of a visualization of those different parts, so that when I come back to it on Monday, it's a little bit of a review and not just all new terminology thrown at you. So we've worked through page 6. We will pick up with the sarcomeres when we come back into class on Monday. And you will have a test mose when you come in, so make sure that you are prepared for that, and I will see you on Monday.